A 24-year-old woman with ovarian cancer presents with high levels of specific microRNAs, miRNAs. The miRNAs are believed to target and silence the expression of a tumor suppressor gene. Which of the following is the most likely outcome of this miRNA-mediated regulation? A. Increased translation of the tumor suppressor gene, leading to tumor suppression. B. Decreased expression of the tumor suppressor gene, allowing for unchecked cellular growth and progression of cancer. C. Inhibition of DNA damage repair mechanisms, leading to decreased mutation rates in the cancer cells. D. Activation of pro-apoptotic pathways, resulting in cancer cell death. E. Loss of expression of the oncogene, leading to decreased tumorigenesis. Answer: B. Decreased expression of the tumor suppressor gene, allowing for unchecked cellular growth and progression of cancer. Explanation: miRNA can regulate gene expression by binding to complementary mRNA sequences, silencing tumor suppressor genes. This results in uncontrolled cell growth and may contribute to cancer progression. A 5-year-old boy with a genetic disorder presents with developmental delays, hypotonia, and seizures. Fluorescence in situ hybridization FISH analysis reveals a microdeletion in the 17p11.2 locus. Which condition is most likely associated with this finding? A. Prader-Willi syndrome. B. DiGeorge syndrome. C. Angelman syndrome. D. E. Williams syndrome. Answer: B. Explanation: a microdeletion at the 17p11.2 locus is associated with DiGeorge syndrome, which is characterized by thymic hypoplasia, hypocalcemia, congenital heart defects, and developmental delay. This deletion can be identified with FISH analysis. Question 3. A 26-year-old man presents with hearing loss and visual impairment. His diagnosis is Refsum disease, which is caused by a defect in alpha oxidation of phytanic acid. What is the most likely result of this metabolic defect? A. Buildup of phytanic acid due to its inability to be degraded. B. Accumulation of homocysteine due to cystathionine synthase deficiency. C. Excessive production of reactive oxygen species in the mitochondria. D. Abnormal glycolysis, leading to impaired energy production. E. Defective methylation of homocysteine, leading to vascular abnormalities. Answer: A. Buildup of phytanic acid due to its inability to be degraded. Explanation: Refsum disease is characterized by a defect in the oxidation of phytanic acid, which results in its accumulation in the tissues. This leads to symptoms such as retinitis pigmentosa, ataxia, and neuropathy. Question 4. A researcher is studying RNA interference, RNAi, and its use in gene silencing. Which of the following types of RNA molecules are most directly involved in silencing gene expression by targeting specific mRNAs for degradation or translation inhibition? A. Ribosomal RNA, B. Messenger RNA, mRNA, C. MicroRNA and small interfering RNA, siRNA, D. Transfer RNA, E. Small nuclear RNA, and C. MicroRNA, miRNA, and small interfering RNA. Explanation: miRNA and RNA are the key molecules involved in RNA interference, a process where these small non-coding RNAs bind to target mRNAs, leading to mRNA degradation or translation inhibition. Question 5. A 30-year-old woman with polymerase chain reaction PCR results showing a frameshift mutation that leads to non-functional protein expression. This mutation was detected in a tumor suppressor gene that regulates cell cycle progression. Which of the following techniques would be most useful for identifying the specific mutation within the gene? A. Northern blotting, B. Western blotting, C. Polymerase chain reaction, PCR amplification and sequencing, D. Microarray analysis, E. In situ hybridization. Answer: C. Polymerase chain reaction, PCR amplification and sequencing. Explanation: PCR, followed by sequencing, is the most effective method for detecting specific mutations in a gene. It allows for amplification and detailed analysis of the gene of interest, identifying frameshift mutations or other genetic alterations. Question 6. A 17-year-old male presents with a family history of early-onset coronary artery disease. Genetic testing reveals a mutation in the LDL receptor gene. Which of the following methods would be most useful for detecting heterozygosity for the LDL receptor mutation in this patient? A. Southern blotting. B. Western blotting. C. Microarray analysis. D. Fluorescence in situ hybridization FISH. E. Polymerase chain reaction and sequencing. Answer E. Polymerase chain reaction, PCR, and seq explanation. PCR and sequencing are the most suitable methods for detecting genetic mutations, such as the one in the LDL receptor gene. Sequencing can identify heterozygous mutations and allow for the precise determination of the mutation's location in the genome. Question 7. A newborn with severe developmental delay undergoes fluorescence in situ hybridization FISH analysis, which detects a microdeletion at the 15q11-q13 locus. This is most likely associated with which of the following conditions? A. Prader-Willi syndrome. B. Angelman syndrome. C. DiGeorge syndrome. D. Down syndrome. E. Marfan syndrome. Answer A. Prader-Willi syndrome. Explanation: the 15q11-q13 locus is associated with Prader-Willi syndrome, a genetic disorder caused by the loss of paternal genes in this region. The syndrome presents with severe developmental delay, hyperphagia, and obesity. Question 8. A patient presents with a history of recurrent respiratory infections, hearing loss, and situs inversus. Genetic testing reveals a defect in the dining arm of cilia. Which of the following conditions is most likely to be diagnosed in this patient? A. Primary ciliary dyskinesia, PCD. B. Cartagener syndrome. C. Marfan syndrome. D. Ehlers-Danlos syndrome. E. Osteogenesis imperfecta. Explanation: Cartagener syndrome is a type of primary ciliary dyskinesia, PCD, characterized by immotile cilia due to defects in the Dynean arms, leading to recurrent infections, hearing loss, and situs inversus.